All right, thanks to the organizers. I'll tell you about some new algorithms that we developed to infer the parents' genomic ancestries from offspring's genotypes. So this is joint work with my collaborators, Iran Halperin, Esteban Borchard, and Sriram Sakuraman. So the motivation for this work really comes from personal genomics. I know many of us here are very interested in personal genomics. What makes it very exciting is the promise of taking the genotype of individual, integrated with various clinical and biological information, and give insights about the individual's past as well as the individual's future. Right, so what does that mean? So to make it a bit more concrete, we would like to take the genotype of individual and give insights of both about the individual's sort of more recent past, like genealogy and family history, as well as maybe more ancient past, such as the population history and the population dynamics and evolution. Going the other direction, we also like to take the individual's genotypes and learn something about his or her disease, future disease risks and disease burdens. So there are, in order to fulfill this very exciting promise of personal genomics, there are many computational challenges that needs to be addressed. Right? So in particular, it's very important to be able to very accurately infer and learn what are the genomic origins of the different parts of the individual's genome. So by that I mean, for example, if we take an African-American individual, right, different parts of her genome is going to come from Africa, come from Europe, or come from Asia. So that's the geographic origin of different parts of the genome. And why is that an important thing to learn? So in order to have a good, accurate inference model about the individual's genealogy and family history, as well as population dynamics, it's very important to have accurate inferences of this, where does each part of her genome come from, right, to have this accurate geographic information. But also, more recently, we've learned that there are many disease risks and disease burdens that are associated with specific ancestral states of the genome. Again, for example, if you're an African-American individual, if you happen to have the African state in a particular loci in the genome, then that actually significantly increases your risk for diseases such as asthma. Right, so having this geographic information of the genome is really a very important link between the past and the future of the individual. And because of this importance, there's been many recent research interest in precisely try to solve this kind of problems, right? So what the current methods do is, given the genotype of an individual, now we can very accurately infer the geographic origins or the local ancestry states of that same individual. So what we try to address here in this paper is an extension of that problem, so it's actually a more challenging problem. So what we like to do is, given the genotype of an individual, we want to learn not just the geographic origin of that individual's genome, but also the geographic origins of each of the parents' genome. Right, so that's our goal, to infer the parents' genomic ancestry from just the genotype of the individual, of the offspring. So why do we want to do this? If we have actually very accurate ways to infer the parents' genomic ancestries, that gives us much better resolution of the population history and of the family lineage. But it also gives us better tools to understand how exactly do these ancestry-related diseases propagate through population and propagate through generations. And moreover, it opens up a window into investigating all sorts of interesting computational social science questions, of which I'll give an application towards the end of the talk. So here's the outline of the talk. So I'll first describe precisely what we mean by genomic ancestry, both at the local and global level. Then I'll define the model and the algorithms that we use to model this genomic ancestries and perform inference. And finally, I'll talk about how we validate the data and some applications. So first, let's just, so we're all on the same page, I want to briefly define what admixture is and what is this genomic ancestry. Okay, so admixture is defined to be the process whereby individuals from distinct populations come together and form families. So here's a concrete illustration of this. Right, suppose we are in some, so the top row here corresponds to some ancestral population, some generations in the past. So that's just for concreteness and for simplicity, assume there are only two distinct population states, right, corresponding to the orange colors and the blue colors. Right? So each individual is represented by these two vertical bars corresponding to the two alleles of that individual. So in the initial ancestry population, there are just two individuals either come from the orange state or from the blue state. So that's focused in on a particular family, a particular trio. Right? So what happens is that the offspring now is going to inherit one ancestry state, one orange state, and one blue state from each of the parents. And then as we go down the lineage, recombination is going to break up the ancestry states and reshuffle them into finer and finer mosaic patterns, right? Where the mosaic corresponds to different parts of the genome. Now it's going to come from different ancestry states. And if you go further down the lineage, towards the present day, the mosaic pattern is going to look more finer and finer, right? You're going to have shorter and shorter blocks until you get to the present day population, where each individual now in this admixed population is going to be represented by this quite intricate pattern of mosaics, where the colors corresponds to different ancestry states. So what do we mean when we say genomic ancestry? So you can either talk about the local ancestry or the global ancestry. So for local ancestry, what we mean is that if you look at this individual and the particular location in the individual's genome, what is the geographic origin or the ancestral origin of that location, right, of that locus? So for example, it can either come from European or African or Native American origin. So that's local ancestry. So global ancestry refers to if you take the individual's genome, what is the overall fraction of the individual's genome that come from each of the ancestry states, right? So how do we think about and model these kind of mosaic patterns that arise through many generations for recombination? So let's focus in on a particular individual. So each individual is going to have actually two distinct mosaic patterns. One's inherited from the mother and one from the father. So first, we just look at the pattern, the mosaic pattern that's current from one parent. There are basically two sets of statistics that are very crucial here that summarizes this mosaic pattern. The first set of statistics corresponds to the frequencies of these different mosaic blocks, right? How often do they occur? The second set of statistics corresponds to the length distributions of these different mosaic blocks, of these different ancestry blocks, corresponding to how long do these blocks are, how long are they, right? So the frequencies and the lengths basically captures very important information about its many generations of admixture. So these are the parameters that we're interested in learning. So more formally, we can think of this as you can also do this with the second parent, right? So the second parent is going to have a different set of mosaic patterns corresponding to two different sets of parameters for frequencies and for the length distributions. So more formally, we can model this as a semi-Markov process. A semi-Markov process is just a generalization of the more standard Markov process or Markov chain. So it's parameterized by exactly these two sets of parameters corresponding to the frequencies and length distributions. So in the previous example, we just have an African-American case study, so there's just two ancestry states. But in general, K is the number of ancestry states, and K can be 2 or 3 or even larger. So just to get some intuition of what this semi-Markov process is doing, let's just go through an example of how we generate a mosaic ancestry pattern from a semi-Markov process. Right, so the first thing you do is that you're given this set of parameters, and you first just roll a die and decide, okay, what is the ancestral state, right? Select it randomly. So in this case, we pick a blue state. And then given that you selected the ancestry state, you sample a particular length from this blue, the length distribution corresponds to the ancestry state. And then you iterate this process. The next state is orange. You pick a length block from there and add it onto your mosaic. And you iterate this until you actually have this mosaic pattern corresponding to the ancestries of this individual. So what makes the computational problem challenging is that for each individual, we actually have these two distinctive mosaic patterns, like two distinctive ancestry chains that's running here, semi-Markov chains, one corresponding to each of the parents. And they can have very different parameters. Of course, we don't actually observe each of these ancestry chains. We don't observe the semi-Markov process. What we do observe are just the genotypes across the genome of the offspring. And from the genotype, what we can do is we can infer locally what is the ancestry state at each position in the genome. Right? For example, we know locally at this position, So here I just put in just to make things concrete, I just put in sort of E for European ancestry and A for African ancestry. Right? So at this particular position, we know that this individual has a European ancestry state and an African ancestry state, but we don't have, so it's unordered, right? So we don't have the facing information, so we don't know exactly where the A African state come from, either parent 1 or come from parent 2. We just observe there's one of each. So so remember that our goal is that we want to learn the parameters of the frequencies and length distributions of both of these semi-Markov processes. Right, so the observations that we have are, from the data, we have these local ancestry states across the genome. What's missing is basically how these local ancestry states are aligned into the parent blocks. Right, so what that means is that, potentially, there could be exponentially many possible alignments into the parent's genomic mosaic patterns that are all consistent with the observed data. So for example, here, both of these are possible scenarios for the two parents that can all be consistent with this set of observations that we observe. And there can be exponentially many such possible scenarios for parents that are consistent with the observed data. So here's, I just want to give some high-level intuition for how the algorithm works. The details are in the paper. So the algorithm, basically, that we call ancestry, basically iterate through a sampling step and an optimization step. Right, so it takes as the input the observed data, which is the local ancestry states of these individuals across the genome. So we initialize the parameters of the two semi-Markov processes. And conditioned on the initial set of parameters, what we can do is that we can actually very efficiently generate exact samples of each of the parent scenarios that are consistent with the observed data. So we can generate aligned parent scenarios that's consistent with the observed data very efficiently. So that's the sampling step. And the optimization step, what we can do is that condition on a finite set of parent scenarios that's consistent with the observed data, we can analytically optimize for the parameters of the semi-Markov process. And then what we do is that we iterate between the sampling step and the optimization step until convergence. So it's similar to the kind of stochastic EM approaches. So just to recap the algorithm part, right? So we take as input just the offspring's genotype. Our algorithm ancestor has two components. The modeling component uses the semi-Markov process. The inference component uses this iterative optimization. What's important is the output of the algorithm. The output is that we have for each parent this set of parameters corresponding to the mosaic patterns, the parameters coming from the semi-Markov process. And in particular, from these parameters, we can actually learn let's say, the global genomic ancestries of this individual, right? The fraction of her genome that comes from African, European, or Asian ancestries for each of the parents. So here's basically how we want to see how well the algorithms work. So we have this a very nice data set, consists of about 230 Mexican trios and 260 Puerto Rican trios. So what's nice is that for each of these trios, we actually have the full genotype of the mother, father, and offspring. Right? So this gives us a gold standard because we can actually explicitly compute what is the genomic ancestries of each of the parents. Of course, we hide that information from the algorithm. We only give the algorithm the offspring's genotype, and we just measure how well are the estimated genomic ancestries compared with the ground truth. So the x-axis here corresponds to the ground truth of the global ancestries. And the y-axis corresponds to the estimations from our algorithm from ancestor. Each dot here corresponds to one parent in each of these populations. And what we can see here is from the scatter plot that we're actually getting quite accurate inferences of the genomic ancestries in both the Mexican and the Puerto Rican populations. So what's nice about the algorithm is that it gives us a lot of information in addition to just the global genomic ancestries. Right? Because we actually have the full parameters of the semi-Markov process, we can also get information such as the block length distributions. Right? The block length is interesting because it tells you information about how many generations has it been since the initial admixture event. So here I'm just showing again, each dot corresponds to an individual. The x-axis, the true block length measured in each of the parents, corresponds to each of the ancestry states. And the y-axis is the estimated block length from our algorithm. And again, for the block length, we are also getting very accurate estimations for each of the ancestry blocks. And I just want to show you how the algorithm compares against a couple of very simple benchmarks, but the benchmarks that are actually surprisingly used in practice. So the table here shows the R-squared values between the estimated genomic ancestries and the observed genomic ancestries. Right? So higher the value, the better. So our algorithm is in the top row, corresponding to ancestor. So the two benchmarks are, one, it's a very simple benchmark, but it is actually used in practice of if you just simply equate the parent's genomic ancestries with the offspring's genomic ancestry. Right? That's the most naive thing that people can do, but it is used. So the other benchmark is you basically make this independence assumption across the loci in the genome. And the columns here corresponds to different populations and for different types of ancestries. And what we see here is that we're actually doing significantly better across all of the populations, all of the ancestry blocks, compared to these, what is currently used. And in the end, I just want to give a quick application of this method. So one of the immediate applications is that now, since we can take the offspring's genotype and learn about the genomic ancestries of both parents, we can now quantify what is the correlation between the genomic ancestries of the couples, right? To get some ideas how much assortment there is in terms of mating patterns based on genomic ancestry. So here we applied it to a data set where we have Mexican populations from 6 different locations in the US. Each dot here now corresponds to one Mexican couple. The x-axis is the one partner, and the y-axis is the second partner, right? The x-axis is the genomic ancestry, the European ancestry of one partner, the y-axis is the European ancestry of the second partner. Just as a quick way to visualize exactly how much correlation there is in each of these locations. And what you see here is that there is actually quite consistent correlations in terms of the European ancestries in these Mexican couples in each of the locations, across all 6 locations. And we can do the same with measuring the Native American ancestries across all these locations. And there's also quite significant correlations in terms of the Native American ancestries across the couples. Right, so this opens up quite an interesting window in which we can actually apply these methods and investigate a lot of interesting computational social science questions about how do these ancestry-related mating patterns, assortment patterns, manifest itself and quantifies these patterns in different populations. Okay, just to wrap up, so here we've described a new method, ancestor, that infers the parent's genomic ancestries from the offspring's genotype. So the algorithm we introduced is based on this model of pooled semi-Markov process, which has a lot of interesting properties. And we validated the method on Latino trios and also discussed some interesting applications. So thank you.
Are there any questions? If you have any questions, there's one.
Yes, I have 2 questions. So if I understand correctly, the input to your algorithm is the alleles at the genotype level from the individual, but already assigned to European or African ancestry. Is that correct?
So yeah, so we just take the general genotype of this individual, unphased genotype you can get from any of the standard chips. Mm-hmm. And the first step of the algorithm is first just to apply one of the standard methods to compute the local ancestry states of that genotype. So in this case, we use LAMP-LD, but you can use other methods to do the local ancestry inference.
And do you compare your block distribution to those predicted by those kind of algorithms like RFMix and others that don't use ancestry family information? They just look at the empirical distribution, and how that compares to those algorithms for, say, for example, for the offspring.
Right. So actually, I didn't describe it in so much detail, but the benchmark that we call independence is essentially, throwing away some of these the correlation introduced by the semi-Markov process, and essentially just look at each of the blocks independently. So that gives you some power in estimating the parents' genomic ancestries, but it actually performs quite poorly, especially in, say, the Puerto Rican populations.
Okay.
Yeah, a similar question, but different angle. How dependent is your algorithm on that initial ancestry assessment being correct. So have you tried to deliberately either, for example, compute an ancestry, leaving out Native American or leaving out one ancestry component, force the ancestry algorithm to make a call, which is probably wrong, and then see what kind of impact that is? Because realistically, there must be some errors in the first process. It would be interesting to understand how those errors affect this your admixed estimation.
Yeah, that's a good question. So in our validations, we've relied on the fact that for these particular ancestries, we do have fairly good reference panels, so we have quite accurate local ancestry inferences. We have tried some simulations along the lines of what you suggested, just to see how robust the algorithm is. So there is actually an extension of the method I haven't described here, where it's more unsupervised. So it basically tries to does simultaneously both the initial local ancestry callings as well as this integrative semi-Markov process. And that seems to be more robust to the version that's described here. So that's something that's work in progress.
So, but errors do propagate. That sort of in the current program, errors do propagate.
Yeah, so if there are systematic, fairly large-scale errors in the initial callings, then that does propagate to the end, yes.
Okay, let's thank James again and move on to the next talk.